number 7. This is one I'm adding that uses a diagram that I have made because there's not a diagram in the book that describes things in the way that I want to describe them. And even though this says Central Dogma Review, and we're not quite done with talking about the Central Dogma, I think it's helpful to bring this stuff in now and then continue on with number 8 about how they discovered what a codon is and that sort of thing. Okay, so just to get you oriented to where we are in this thing. This is a eukaryotic cell. Later on in chapter 3, we'll talk about prokaryotes versus eukaryotes. For now, you can think of prokaryotes, you can think of bacteria as an example of a prokaryote, and you can think of us, humans, as an example of eukaryotes. So in eukaryotes, they have a nucleus. Prokaryotes don't. Bacteria don't have a nucleus. And we're going to be talking mostly about how things work in eukaryotes. So here's the edge of the nucleus, and this whole thing here is considered a cell. So this is what happens inside the nucleus, inside this little line here, and then outside of that is the cytoplasm. This is the liquid part of the cell where things are floating around, and you're going to watch a video later that happens mostly in the cytoplasm. Okay, in the nucleus, that's where the DNA is. This is again talking about eukaryotes, or this would be anything higher than bacteria. And the DNA is in the nucleus. It's wrapped up in protein, and how the DNA is accessed and what parts of the DNA are read is pretty complicated. So I say genes are complicated. Control regions can be far from the protein coding regions. The part that actually turns into protein that is read by a ribosome is only part of the gene, and other parts of the gene that says, okay, let's make a lot of this gene or let's not make much of this gene, those can be physically far away on your DNA strand. And this hash mark 3-armed thing is a protein complex that when the DNA gets read, it might grab things that are right next to the part that is going to turn into protein, but also maybe far away. And I just want you to know that that is complicated, and the part that turns into protein is not the whole thing. Okay? If we zoom in on the DNA and we look at the base pair sequences, so remember we talked about adenine, cytosine, thymine, and guanine as a base as part of a nucleotide. The nucleotide is the 3 phosphate groups, the sugar, and the base. So the thing that is different is the base, so we just use the base as a shortcut for talking about these nucleotides. And remember, there's a 5' end and a 3' end, and a double-stranded piece of DNA is anti-parallel. If it's 5' on this end, then the other strand has got to be 3' on that same end. And same over here. If it's 3' on this end, then the strand that it's bound to has to be 5'. And you have standard base pairing, and then it's A with T and C with G, because these are 2 strands of DNA. So one of these strands of DNA is going to be read by RNA polymerase in the process of transcription. So we just talked about this in the last podcast. But which one? Do I read this one on the top, or do I read this one on the bottom? It turns out that it matters. So the mRNA that is produced from this is going to be in the sense direction, and that essentially means 5' to 3' left to right, like you're going to read a sentence. 5' would be on the left-hand side, 3' on the right-hand side. That's how the ribosome is going to read it. And in order to make that, you have to read it from this bottom strand, which is going in the opposite direction. So all nucleic acids, 2 strands DNA, or a strand of DNA that's turning into a strand of RNA have to be anti-parallel. So in order to get your mRNA to be in this direction, you have to read this antisense strand, okay, which is also called the template strand. It acts like the template to make mRNA. So the other strand is called the sense strand or the non-template strand. The antisense strand or the template strand is the strand of DNA that is read by RNA polymerase. And when it's read, it is converted into RNA. So you remember that RNA does not have a T, instead it has a U. So T binds with A, okay, G with C, no problem there. But A would normally bind with T if it was DNA, but now this is RNA, so now A is going to bind with U. C with G, T with A with U, and so on. And so I get an RNA copy of this DNA, and is copied exactly as according to the sequence on the antisense template strand of DNA. Okay? Now, that mRNA has got more sequence than will be used by the ribosome to turn it into protein. It has small, not necessarily small, but it has stretches of sequence that are intervening the parts that are going to actually turn into protein. So there are these two things are called exons, EXONS, E for exon, and introns, I for intron. The introns end up getting cut out of the sequence before the ribosome reads it, and the exons stay in. So this thing here that is exact copy, exact RNA version of the DNA, that's called a primary transcript. And then it gets processed while it's still in the nucleus. We're still in the nucleus here. And when it gets processed, the exons get joined together and the introns get cut out. Okay? Introns get cut out, exons stay in. So one of the things that is kind of amazing about our DNA is that we can make more than one version of protein from a single gene. So we think that there's about 200,000 or 300,000 proteins in humans. That's an estimate. We're still working on that number. Scientific community is still working on that number. But we know from sequencing the human genome that there's about 22,000 genes. So from 22,000 genes, you get about 300,000 proteins. Obviously, you must have more than one protein per gene. And here's one of the ways you can do it. So this gene here has 3 exons. If I put just 2 together to make an mRNA, I get one version of a protein. If I put just the third one together in an mRNA, I get another version of the protein. So I can mix and match these exons and put them together into an mRNA to be read by a ribosome in this processing part of the sequence. This is after it's been transcribed by RNA polymerase. And then when you cut out the introns, you can put together different exons and you get different mRNAs from that. Okay? So you get more than one processed mRNA from one gene, typically. Okay. So now you've cut out your introns and you're left with your exons, and that leaves the nucleus. This thing here is a nuclear pore, and it leaves the nucleus, and now it's out in the cytoplasm where it can be worked on by a ribosome. And in the ribosome, we talked about ribosomes in the last podcast. There's a small subunit and a large subunit, and they come together around the mRNA that has been processed and start translating protein. So how does the ribosome know how to put amino acids together in what order? It reads that order from the mRNA, and it starts on the 5' end of the mRNA, and the ribosome actually moves toward the 3' end as it reads it, and we'll see a little video of that. Okay, on the mRNA, again, remember it's RNA, it's right in the name, and so there's no T's, there's U's instead. And on that mRNA, someplace near the 5' end, there's the first codon, and a codon is a group of 3 nucleotides, and 3 nucleotides are required to code for 1 amino acid. Okay, that's really important. 3 nucleotides is a codon. 1 codon codes for 1 amino acid, and 3 nucleotides makes up 1 codon. Now, the very first codon for almost all proteins, we'll just say for the sake of this class, all proteins, the very first codon is AUG. That is the sequence of the first codon. That codes for a tRNA that's going to bind to that codon that has the anticodon sequence that'll bind to it. So A with U, C with G. So it is anti-parallel to this mRNA. This mRNA is going 5' on the left, 3' on the right. The tRNA that's going to bind to it is 5' on the right and 3' on the left, anti-parallel. And these base pairs match up, and that first codon has, that tRNA has a methionine stuck to it. So the first amino acid will be methionine, the first codon is AUG. And that first codon is near the 5' end, but it's not exactly butted up against the 5' end. There's some nucleotides before that. They're called untranslated region, UTR. So the mRNA is bigger than just the codons. It's got some untranslated region at the beginning of the sequence and also at the end of the sequence. All right, the mRNA then sits in the ribosome. The ribosome scans along and looks for this first codon, AUG, and when it finds it, the tRNA from the methionine pops into this site called the P site and has a methionine stuck to it. Then the next codon, in this case it would be AUU, but it could be anything, okay? The next codon, you're going to read 3 at a time. It'll be AUU. tRNAs bump in and out of this A site trying to make a match to AUU. It would be an anticodon that would be UAA, okay? And if there's a match, what happens is the methionine from the first codon is transferred over to the new amino acid, and now a peptide bond is made between those 2 amino acids by the ribosome, and then now your protein is 2 amino acids long. Now the whole ribosome moves toward the 3' end. What was in the A site moves to the P site, and what was in the P site goes to the E site. So the P site had its amino acid taken off of it. Now it's just an empty tRNA, and it gets ejected. E for eject. Okay? And now this whole 2 amino acid thing gets moved to the P site, and now the A site is empty, and you can read the next codon. And it keeps on chugging along like that toward the 3' end, 1 codon at a time, until it hits what's called a stop codon. And the stop codon codes for the ribosome to stop making protein instead of 1 more amino acid. Instead it says stop making this protein. When it runs into that stop sign coded for by a stop codon, it essentially takes apart the whole machinery. So the large and the small subunit come apart. The protein that you were making gets cut off the ribosome, goes off and folds up to do its job as a protein, and then the ribosome can start over on a new mRNA. Okay, so one other important thing that is important for something that will come later on in the class, and that is, what about mutations? Okay, so When you are reading an mRNA, you read it one codon at a time. One codon is 3 nucleotides. So I'm going to read these 3, and then these 3, and then these 3, and so on. And that 3 at a time is called a reading frame. So I've got a little frame, a little bracket here that I'm going to read 3 at a time. These 3, then these 3, then these 3. So you can think about it almost like reading words in a sentence. And if all of your words have 3 letters in them, the sentence makes sense. The big red fox ate one bug. Okay, and that's not great literature, but at least you can sort of figure out that it makes sense. But if I have a mutation, let's say I have a mutation and I get rid of just one letter in the sentence, the ribosome is still going to read 3 things at a time. It just shifts down its frame. And now it's reading these 3, and then these 3, and then these 3, and all of a sudden this sentence makes no sense at all. And that sort of thing can happen inside a cell if there's a mutation such that the mRNA has a deletion in it. It can shift the reading frame, and so now it is not making the protein you intended to make, but instead makes a protein that doesn't make sense. That's called a frame shift mutation, and it's almost always a bad thing for the cell that is making that protein, because now it can't make a protein that is going to have the right shape, and if proteins don't have the right shape, they can't do the right job, and usually that protein just degrades and you don't get that protein. So there's some very famous frame shift mutations in human biology. We'll talk about those later on in the class. But this diagram is posted on Brightspace, called Central Dogma Review, and it's a good review for the many things that we're talking about in the Central Dogma, where DNA is transcribed into RNA, and then RNA is translated into a protein.